All right, everyone, welcome back. Let's get things started with Chapter 1, An Invisible World. First, our objectives. After this chapter, you should understand the scope of microbiology, the scale and diversity of microbes, and the role of microbiology in our lives. Before we begin, let's talk about the definition of microbiology. So, biology has in it bio, which stands for life, and ology, which is the scientific study of something. So, this is the science that studies living organisms. And micro essentially just means extremely small, usually so small it cannot be seen without a magnifying aid. So, microbiology essentially studies microorganisms or microbes, which are organisms that cannot be seen with the unaided eye. This term encompasses a wide diversity of things if you think about it. Microbes can be unicellular organisms or multicellular organisms. They can be prokaryotic or eukaryotic, which we'll talk about later in this chapter. They can be heterotrophic or autotrophic, and that basically has to do with their types of metabolisms they can do. They can be life sustaining organisms, and these can be organisms that make different foods for us or keep our microbiome healthy, or they can be life threatening. As we'll see, many of these organisms can cause diseases. In a way, we are made of microbes because even our cells that make up our body are extremely small living things that cannot be seen with an unaided eye. So, as we just alluded to, our microbiome is made up of microorganisms as well. And so are the cells that compose our multicellular bodies. It might not seem like it relative to our own size, but microbes span a wide range of sizes. If we look at this microbe scaling chart, we see a period character in Helvetica, type 12 point size, is about 500 microns or micrometers right here. Um, the largest organism we're going to be dealing with is the amoebas or protists. They're a type of protist. And they can be up to 300 or micrometers in size. So some of these can be barely visible to the naked eye. By the way, the naked eye just means you cannot see them without the aid of a microscope. As we get progressively smaller protists, then we have the paramecia. Okay. Um, the paramecia are about 250 microns in size, or about half the size of a period. Um, and then we make our way down to even baker's yeast, which are about 10 micrometers in size. So you could fit 50 baker's yeast in a Helvetica period. And down here at the bottom, we start getting into bacteria with Escherichia coli, hereafter referred to as E. coli. E. coli is basically the lab rat of bacterium. It's the most well studied bacterium we have right now. It's found in the digestive tracts of humans and other animals in the intestines. It's shaped like a pill and is about 2 micrometers or microns in size. So 250 to 500 E. coli can fit in a period. We most certainly cannot see this organism with the naked eye. So as you can imagine, almost all of these require a microscope to visualize. So let's continue here. We have other types of bacteria, lactobacillus, at about the same size as E. coli, 2 microns by 1 micron, same sort of shape as well. We use lactobacillus to make foods, such as yogurts and other fermented products. Then we have cyanobacteria, and these are a group of bacterium that are photosynthetic organisms. In other words, they can use sunlight in order to create energy or harvest energy. We'll get into cyanobacteria later in the semester. Staphylococcus are bacteria that are found on your skin. Most are harmless, but some can cause disease. Uh, these cyanobacteria are round in nature and they're about 1 micron all the way. Now, as we can progress even smaller, the next step down are viruses. Viruses are a much smaller level of size altogether. So here we are still hitting about the micron size with bacteria, and viruses take a big leap down, about 1/3 the size. The smallpox virus is about zero point 0.3 micron, continuing down to 0.03 microns in size are the rhinoviruses. And so you could fit over 16,000 viruses in the volume of an amoeba. So this gives you a glimpse as to how diverse microbiology is in its scope of study. We will study at an extremely diverse group of organisms in this class, so much so that this class really only scratches the surface of what we know about microbes. In fact, a study done by Indiana University in 2016 predicted that there are a trillion species of microbes, 99.99% of which have yet to be discovered. So, of course, there must be a system for keeping track of all these microbes and living organisms in general. And how we do that is we classify the living organisms into different groups. We can group organisms from larger, more encompassing groups, such as kingdom, down to species. Here you can see the organization goes from kingdom to phylum to class to order to family to genus to species. For humans, that would start with Animalia, Chordata, primates, Hominidae, Homo, and Sapiens. Each one of these groups, the members within that group become smaller and smaller. The genus and species name is the full name given when referring to an organism. So we are Homo sapiens. And Escherichia coli is a commonly studied bacterium where Escherichia is the genus and coli is the species name. Those always go hand in hand with each other, and that's called the binomial naming system. The naming of microbes has a certain convention, and this is very important for you to follow precisely. The entire genus and species name is all italicized. The first letter of the genus name is capitalized, and everything else in the name is lowercase. Examples of this are Escherichia coli, notice all italics. Here we can see the whole name is italicized, and the first letter is capitalized. In writing, when you're writing these, of course, you want to make sure you say the full name the first time. Each subsequent use of that name, though, you're going to abbreviate the genus by just writing the first letter and capitalize it, period, and then write the species name. And you're going to do this in your own writing as well when you do your writing assignment for this class. And I want you to make sure when you're using your whatever software it is that you're going to write the report on, to be careful that the autocorrect does not capitalize the C in coli. And if it does, make sure you change it back to a lowercase letter. Another example is Saccharomyces cerevisiae. And this is a very important organism to us as well. It comes from the root words myces, which means fungi, and saccaro, which is sugar, and cerevisiae, which means beer in Latin. And just like the Spanish word cerveza means beer. So this is a sugar-eating fungi that makes beer. And finally, we have Salmonella enterica, and it is named after Dr. Daniel E. Salmon, and enterica coming from the Latin word for intestine. Checkpoint 1. Remember to submit your checkpoint question assignment online under the checkpoint question assignment for the chapter. You will not see the question rewritten there, so take a moment to pause the video if you need to, write down the question, and then the correct answer for it. So checkpoint 1 is, what is the right way the following name is written? Okay, whichever answer you select, make sure to record the letter with it because you'll actually choose the letter when you go to submit your answer, and it won't give you the possible answers here. Let's get into the different types of microbes. Microbes are highly diverse genetically. In fact, there's more difference genetically between E. coli and Salmonella enterica than between humans and dogs. They are also diverse in their metabolism, behavior, and phenotype. We have bacteria, we have archaea, fungi, protozoa, algae, viruses, and helminths. And we're going to try and briefly cover each one of these microbes. Now, this is not an exhaustive list of microbes, but these are microbes that we will be covering in class. Let's first discuss the traits of bacteria. Now, all of these traits we will give much better explanations later, but for now we will summarize them. All bacteria are unicellular, and that means they don't form a complex organism of more than one cell. Bacteria can form a complex community with other bacterium, but they're still considered unicellular organisms that don't require multiple cells in order to function and be alive. They are also prokaryotic. Most of you should already know what prokaryotic means by now, since you should have taken a high school biology course or some college courses. And if you don't recall, a prokaryotic cell basically means that their chromosomes are not housed in something called a nucleus. Prokaryotic means pre-nucleus. A nucleus is an organelle inside of the cell that is a membrane limited area. And so what that means is inside of the membrane of a nucleus is where the chromosome would be located. For bacterium, they have a region in the cell you can find their chromosome, but it isn't surrounded by a special membrane. They reproduce through binary fission, okay, which is a type of splitting of a cell into two equal daughter cells, and it's not sexual reproduction. So they do not require male or female or other sex types in order for this reproduction to occur. Chromosomes are the form of DNA found in cells that store genetic information, called genes. And bacterial chromosomes are kept as small circular chromosomes without a beginning or end. And as you should recall from previous biology courses, that's not the case for organisms like us humans. We have linear chromosomes where there's two ends to the chromosome, the beginning and the end there. Additionally, their cell walls contain a compound called peptidoglycan. Their metabolisms are very complex and they can function in a variety of ways, so they can be heterotrophic or autotrophic. Those terms define what they eat in order to get their carbon to build cell materials. Heterotrophic organisms have to eat something else that's already made organic molecules or molecules made of carbon. So, an example of heterotrophic would be humans. Humans are heterotrophic. That's all we are. Okay, we don't have the ability to go out and get our own carbon from carbon dioxide and turn it into biomolecules. We eat plants and animals and fungi, things like that, to get our organic compounds. Autotrophic organisms can take CO2 from the air or other what's called inorganic carbon that's not already turned into biomolecules, to create organic molecules from it. The example that we're all familiar with for autotrophic organisms are algae and plants that breathe in CO2 to make their organic compounds. An example in bacteria are cyanobacteria. Lastly, many bacteria can swim with the use of a flagella. So if you look at this organism here, it kind of has a mouse tail that is called the flagella. And this mouse tail here at the base, it can spin and rotate around. And this rotating around whips the flagellum around and allows locomotion to occur. Let's review peptidoglycan here shortly. Peptidoglycan means short proteins, peptides, and sugar or glycan. So it is a molecule made up of proteins and sugar molecules. And there is a lot going on here so just bear with me. But if you look here, we have two different types of bacterium. We can have a gram-positive and a gram-negative bacterium. And this image was taken from OpenStax, but these G's really should be capitalized. Gram is named after a scientist who made this categorization here. So since it's named after the scientist who studied peptidoglycan, it should be capitalized because it's a name. But as you can see, there are several compounds you need to be aware of. So we look at this bottom of this caption here, we'll see that we have an acetylglucosamine, okay, and an acetylmuramic acid. And these are the N-acetylglucosamine are the little blue spheres, and the N-acetylmuramic acid is signified by the yellow spheres. These two compounds are sugars and are connected to each other through peptides, okay, which are short amino acid chains. If you do not recall what amino acids are, please review the basic four biomolecules of life from your prior biology course or do a little research there. Again, we have these N-acetylglucosamines and acetylmuramic acids, and these are derivatives of carbohydrates or sugars. They're basically sugars. Gram-positive bacteria usually have a much thicker peptidoglycan with many more layers of peptidoglycan, whereas gram-negative bacteria has a very thin layer of peptidoglycan. If you look at the peptides of the gram-positive peptidoglycan, you will see a pentapeptide bridge. And so um, both the gram-positive and gram-negatives have these uh, tetrapeptides, and in gram-positive bacterium, they have a pentapeptide bridge, which connects one tetrapeptide or four peptides to another four length long amino acid peptide bound together with a pentapeptide, which is a protein that has one, two, three, four, five amino acids in it. And this helps to reinforce the peptidoglycan layers there. Okay. In gram-negative bacterium, the tetrapeptides are just directly linked to each other. While we're talking about bacteria and their cell walls, let's discuss a little bit about their shapes. Shape is very important because it's often the first step in identifying an unknown pathogenic bacterium or any bacterium. There are three main different shapes in bacteria. There is coccus, which is Greek and it means spherical or ovoid. Okay, like in this first example, the green bacterium on the left-hand side of the right image, you can see that they're kind of ovoid in shape here. If you look carefully, you can see a little invagination here on this bacteria, these bacteria here. And what that is these bacterium are going through the process of cellular division. Remember, it's called binary fission. And so this is a little septum that's forming here, and these two are eventually going to pinch off. You can see it occurring a little bit better in this pair here in order to become their own unique coccus bacterium. And that's just a fact that you're going to see with many of these images of bacterium because they're actively growing and dividing. So a coccus cell might often come in pairs, or it might even come in several chains linked together or grape-like clusters. And we'll discuss what that phenotype is in a moment. The next shape is bacillus. And bacillus means, well, they kind of look bacteria shaped or sort of pill shaped or rod like. Okay, and that's going to be the middle image here. So this is an example of a bacillus bacteria. And some bacteria are actually also named after their phenotype or their shape. So we can have a streptococcus pneumoniae. That's an organism that causes pneumonia. Or a bacillus cereus. This is a soil bacterium. Okay, and bacillus is in its name because it's shaped like a bacillus bacterium. The final shape is spiral, and they have a corkscrew shape. And here we see in this last image a corkscrew shape bacterium. So for now, that's enough about bacteria. Let's discuss archaea. In the image, you can see Loki archaeum. These Loki archaeum are the closest known living relatives to eukaryotes, a group that includes animals, plants, fungi, and others. Archaea are unicellular as well. They are also prokaryotic, so they lack a true nucleus. They have more like a nucleoid region, so they don't have a membrane bound chromosome. They also reproduce by binary fission, and we get to see some great examples of binary fission in that previous slide, which is an asexual form of reproduction again. The prefix letter A just means without. So being asexual means they are without sexual reproduction. They also have circular chromosomes. So they're looking very different, they're looking very similar to bacteria, but they're not bacteria. They're very different. And in other ways, we'll get to later in this course, they're very similar to us. They are genetically something in between eukaryotes and bacteria. More on that in a moment, and we'll get to that much later in the semester as well. One way they differ from bacteria is that the cell walls, for example, do not contain peptidoglycan. And these little dudes often live in extreme environments. So they're often categorized as what we say are extremophiles. The word philes means lovers and extreme environments. So they're lovers of extreme environments. And also, as stated in the textbook, they are not known to be direct causes of diseases. There are three main groups of archaea. There are the methanogens. The root word gen means to generate. So this means that they generate methane during their carbon metabolism. We have the extreme halophiles, which halo means salt. So we have salt loving and we mean it. They live in extremely salty places like salt flats. And next we have the extreme thermophiles. And in this case, thermo does not mean cold, it means hot. These are hot loving, and they live in hot, often sulfurous water, such as hydrothermal vents in the bottom of the ocean or natural geysers. Now, I have this article here because there are often many exceptions in biology. This is one of them. So, if you remember earlier, I said that from the textbook and many other textbooks and sources, they say that archaea are not known to cause diseases. And this is why it's important to pay attention to the lecture material and not outside sources. I want you to have a basic common understanding of these things, a shared understanding. Okay. So, um, again, these sources will say that archaea are not known to cause diseases. But we're starting to punch some holes through that hypothesis. So, this paper is a review article about how methanogenic archaea that live in our intestinal tracts that produce methane, which itself might be a gastrotransmitter. It says archaea are the only confirmed biological sources of methane in nature, and Methanobrevibacter smithii is the predominant methanogen in the human intestine. And this is linked and has a strong association between delayed intestinal transit times. In other words, the production of methane may cause constipation. However, let's be cautious with this article just because it's an observational study and it isn't clear if it is just a correlation or an actual causation at this point in time. Let's jump to fungi. We're getting a little bit weirder and complex in cellular organization with fungi. They can be both unicellular or multicellular. So, obviously, not all fungi are microscopic on the multicellular level. They are what we call eukaryotic. So, they contain a nucleus. Eu means true, and karyo means nucleus. So, they have a true nucleus that has a membrane that holds their chromosome. They can reproduce asexually or sexually. Their cell walls do not contain peptidoglycan, however, they do contain chitin. Chitin is the same substance that forms the exoskeletons of many insects. It is a long chain of N-acetylglucosamine, the same sugar derivative that we discussed in peptidoglycan. They are heterotrophic, so if you remember, hetero means that they're getting their organic compounds from something else. Different, so it's different than them, right? So they can't harvest the CO2 themselves or the carbon. They do not do photosynthesis. I will make sure I point that out because it's easy to be fooled because they are green, but they don't photosynthesize. Examples of fungi include molds or yeasts. And remember, we discussed Saccharomyces cerevisiae. That was the fungi that eats sugar and makes beer. Yes, that is a fungus. So the yeasts that are used to brew beer and wines, etc., are unicellular fungi. Now we've arrived at protozoa, so we're getting a little bit bigger here. Remember that these were the big boys on the block, being as big as 300 microns in size, and a period is about 500 microns in size. Protozoa are unicellular or single-celled, and they are eukaryotic. They can reproduce sexually or asexually, and they can be free living. That is to say that they don't live inside of another organism, or they can be parasitic, living inside a host. For example, the Plasmodium falciparum that you see in the upper right is the protozoa that actually causes malaria and is transmitted by mosquitoes. Amoebas are mostly free living, and they can be heterotrophic or autotrophic, and will be the largest single celled organisms we study in this course. Algae are typically thought of as being unicellular, although they can be multicellular as well. I'm not going to have that one on the exam just because the textbook says one thing and I don't want to confuse the topic further. Not all algae are technically microbial, and that's obvious for anyone that's gone and ate sushi before because what do they roll up your sushi with? Kelp. Kelp is a type of algae and it's multicellular. They are eukaryotic and they can reduce sexually or asexually. Their cells are composed of cellulose, which is a carbohydrate that is organized in a special way that makes it durable and semi rigid. They are autotrophic, so they photosynthesize and they capture their own carbon through CO2. This is called CO2 fixation because the carbon in that CO2 is fixed into organic compounds such as cellulose and sugars and other compounds. In the image to the right, we can see the food product spirulina, which is technically not an algae but a bacteria capable of photosynthesizing. These bacteria are called cyanobacteria because they are bacteria that are bluish green or cyano in color due to the pigments used for photosynthesizing. They are often miscategorized as algae, but far from it. In fact, they aren't even eukaryotic organisms. All right, now we are getting really small. We are going to look at viruses. Viruses are extremely small, even for a microbe. They are acellular, in other words, without a cell. And we're going to talk about this quite a bit more later, but they actually are not made up of a cell at all. Although if you look at this image of a human papillomavirus, it sort of appears like a cell. They are often pathogenic to humans. Although there are so many viruses, most all of them are not specifically pathogenic to humans. Crucially to remember, they require other life forms in order to fulfill some of their basic functions. So because of this, they are not really alive, they cannot reproduce or metabolize independently. Rather, they serve more as a source of genetic code that hops from one living organism to the next. To a degree, they act as some sort of a shuttle for this genetic information. In order to protect that genetic information until it finds a new host that can metabolize or reproduce for it, they are composed of genetic material DNA or RNA that is surrounded by a protein capsid. And we will never see viruses using a typical light microscope, as you commonly see in labs. Uh, the image you see here on the right is done with what's called an electron microscope, a very expensive piece of machinery used to see things very small. Helminths are a whole different horse amongst themselves. We're getting a lot more cellular complexity with these helminths. They are multicellular, and there's a lot more specialization between the cells in this organism. Helminths are animals, and as such, they are multicellular and they have nervous systems. They are only microscopic during certain life stages, and they are quite horrific. Some of them, so if we look at this hookworm, for example, here on the side, they can enter the host by burrowing through the skin itself. Hookworms are common in the southeastern part of the United States and can be transmitted by people walking barefoot on soil. Their microscopic stages include eggs and larvae. Checkpoint 2. B. anthracis is the bacterium known to produce the toxin anthrax. What shape does B. anthracis exhibit? Note that there are many bacteria that form each strand here. Checkpoint 3. Recent satellite data indicates that there is a large amount of extremely salty liquid water underground on Mars. If this water were inhabited by a microbe from Earth, what type of microbe would it likely be? Checkpoint 4. An unidentified microbe is found to reproduce asexually, contain a nucleus, and possess a cell wall made of chitin. What type of microbe is this? So, why would anyone want to study microbiology? One important role that microbes play is in the process called element recycling. Microbes are the only type of living organism that can capture elements such as atmospheric nitrogen and convert it into a form that is useful for all other organisms. See, nitrogen makes up about 70% of the gases in our atmosphere. And this is the case because it is an extremely stable compound that is very hard to oxidize or turn it into something usable by the other microbes. It comes in the form of dinitrogen, which is two nitrogens attached to each other. And this form is very hard to break the bonds between the two nitrogens. However, nitrogen is one of the four main ingredients that comprise biological organisms. We need it because there's an essential biomolecule called amino acids and that makes up proteins. And so living things need this nitrogen in a usable form in order to survive. And there are certain microbial species that are the only living organisms that can convert atmospheric nitrogen or dinitrogen into a usable oxidized form for amino acids. Addition to amino acids are also nucleic acids that have nitrogens in them. And these are the materials that store our genes or genetic information. If you think about humans, we can breathe in nitrogen, but we can't consume it to make amino acids. We're not breatharians. We consume products that have proteins in them, and we break those down into their constituent amino acids. We use those amino acids to build our building blocks. This is also the reason why you often use nitrogen fertilizers, because the nitrogen fertilizers contain some sort of oxidized form or some sort of form where the nitrogen is fixed from its atmospheric form. For this reason, it's really hard to get enough nitrogen into the soil. Another thing we use microbes for is sewage treatment and bioremediation. And this is where microbes can break down contaminants and toxins found in sewage or waste from industrial processes in order to make them more environmentally friendly. Microbes are essential for producing a lot of our fermented fruit, foods, and beverages. This includes yogurt, pickles, sauerkraut, beer, wine, chocolate, the list goes on and on. They are also very important in newer forms of biotechnology. Besides food biotechnology, um, these are newer processes that include genetic engineering and medical usages, such as producing human insulin. We produce human insulin typically in yeast or animal cells, in cell cultures, in test tubes or vials. Additionally, they are also used to make industrial products such as biofuels, plastics, food additives, polymers for material biosciences, and for providing gene therapies for humans. Microbes are very important to us in our human microbiome. The human body is composed of about 30 trillion cells, and it is believed that they cohabitate with about a 1-to-1 ratio to the microbes in our body. So there are about as many microbes as there are human cells. So perhaps the most intimate way that microbes affect human welfare is through the human microbiome. Imagine if you took away all of the human cells in the human body of the human body and leaving just behind the microbes. Observing just the microbes, you would still be able to navigate the different regions of the body based on what microbes and in what ratios of those different microbes are present in each area. Even areas like your elbows have a distinct community of microbes versus your navel. Microbes are essential for digesting our foods. They provide us with vitamins and they within our digestive tracts, and they prevent other pathogenic microbes or invading microbes, the bad ones, from taking up residence. They also are believed to be involved in certain signals to our brain and to the rest of our body, and therefore contribute to the regulation and function of our bodily systems. The human microbiome is the set of microbes that live stably in and on the human body. The microbiome is seeded at birth and helps maintain our health throughout our lives. Here's an interesting study on this topic, the title of which is The Partial Restoration of the Microbiota of Caesarean Born Infants via Vaginal Microbial Transfer. The article shows that babies that are born of a C section are underrepresented in certain microbial populations. And this might be due to the fact that not being born vaginally, they aren't exposed to the microbiome of the mother. And that by essentially swabbing newborn children birthed via C section from a sample taken from their mother, it can help to restore these populations at birth. This was an interesting paper that came out about building the bio-based economy and biotechnology. The process begins with feedstocks. This could be biomass, which could be a foresting byproduct from the logging industry. It could be algae that's harvested from the oceans, or methane released from waste management sites from off-gassing methanogens, as well as many other different sites or sources. And we feed those industrial biotech, those cultures, these feedstocks to grow specialized microbes. We grow big vats of a culture. But instead of growing cultures of yeast to make beer, for example, we're growing microbes to make something else other than alcohol. This involves some sort of synthetic biology where these microbes are engineered with different enzymes to make different products. And then you go through a biorefining process. And you might need further gene editing of these microbes, but ultimately we're building products directly from microorganisms. There are many different products we can make. We can produce sustainable fuels, or we can make industrially relevant chemicals. We can make different polymers, such as plastics and other coatings, and things that are used in industry, such as food ingredients. So, just as we can make foods, we can just as well make ingredients for other foods. This is very big business. I think a lot of people would be surprised by the numbers. So, here we're going to take a look at revenues for the US, uh, for the companies in the United States in 2012. And if you look at this pie chart, it's broken down to three categories. We see red biotech. Red is just a color that symbolizes what are called biologics. And these are basically complex organic molecules made from living organisms, from microorganisms, such as insulin, as we've previously discussed, or it could be some antibodies that are produced, or a whole slew of different things like gene therapies. This has a $91 billion revenue in the United States, which is enormous. That's per year. GM crops are another one, and they're not really microbial in nature. They can be, but for the most part, this is like your GMO corn and soy, etc. And it's a $128 billion business in the United States. Next, we have industrial biotechnology. I think this might be surprising for some who think that medicine is kind of the blockbuster for revenues. But actually, industrial microbiology brings in over $105 billion in annual revenues in 2012. It's also the fastest growing industry amongst the three. And all in all, there will be a lot of very exciting things to discuss in this course and a lot of exciting things on the horizon in microbiology. So that wraps up today's lecture. We will finish up chapter 1 in the next lecture. Thank you all for being patient and listening to the video, and I hope you will join me in the next round. If you have any questions, feel free to email me, and also remember to submit your answers through the quizzes portal on Canvas for the checkpoint questions. Thank you.